Hello everyone. This time we will talk about section 4, cell death. Well, we meet this page again. As we know, when the cell encounters severe or persistent stress or the stimuli are inherently harmful, cells undergo death. There are two forms of death, necrosis and apoptosis. I will introduce necrosis first. It is local cell death in living tissue. Necrosis is the main mode of pathological cell death. Mostly are developed from reversible injury. The morphology of necrosis includes changes of nuclear, cytoplasm, interstitial tissue. The most important morphological features of cell necrosis is in nucleus. There are three patterns of nuclear change: the pycnosis, karyorrhexis, karyolysis. This picture shows us different patterns of nuclear necrosis. The first one is normal cell. We notice here is the nuclear membrane. This is nucleolus. The first pattern is pycnosis. The nucleus shrinked and has increased basal failure. DNA condenses into solid shrunken mass. The second pattern, karyorrhexis, because of the disintegration of nuclear chromatin and the rupture of nuclear membrane, nuclear fragmentation occurred. It broke into several pieces, just like here. The third pattern is karyolysis. In this pattern, non-specific DNAs and protease activate, decompose nuclear DNA and nuclear protein. The dead nucleus will disappear within 1 to 2 days. It should be noted that the pycnosis, karyorrhexis, and karyolysis do not always happen in this order. Here is a diagram showing us the sequence of occurrence. A normal cell can go to karyolysis directly or goes through karyorrhexis and karyolysis process. There are various orders. Let's move to the cytoplasmic changes. Because of the decrease of ribosome glycogen granules, the increase of cytoplasmic denatured protein, the cytoplasm becomes eosinophilic and homogeneous. In the picture below, the first picture is normal cardiomyocyte. The second picture is necrotic cardiomyocyte. We can see here the myocytes bend and break and are darker red in color. Sometimes, when the enzymes digest the cytoplasmic organelles, the cytoplasm becomes vacuolated and appears to be moth-eaten. Dead cells may be replaced by large whorled phospholipid masses called myelin figures that are derived from damaged cellular membranes. The third picture here shows us the myelin figure under changes of stroma. Stroma is more resistant to injury than parenchyma. The collagen fibers undergo swelling to lysis, finally disappeared. We also need to understand that cells' contents often leak out in necrosis, trigger inflammation and digestion of dead cells by lysozymes. Necrosis includes many types: the coagulative necrosis, liquefatic necrosis, gangrene, caseous necrosis, fat necrosis, fibrinoid necrosis. The first one is coagulative necrosis. It occurs in all solid organs except brain, such as spleen, kidney, heart, usually in the situation of infarct. This is a gross specimen of kidney. The focus is here. Gray or yellow-white, with clear boundary and firm texture. Under microscope, The component cells are dead, but basic tissue architecture is preserved. This is glomerulus. This is renal tubules. We can still recognize the organ as a kidney, although the cell is dead. That's because coagulative necrosis denature the structural protein and enzymes at the same time, so the proteolysis of lysosomal enzymes are blocked in the dead cells. The second type, liquefatic necrosis. It is seen in focal bacterial infection, occasionally in fungal infection. Microbes stimulate the accumulation of inflammatory cells and the enzyme of leukocytes to digest the tissue. Tissues turned into a liquid viscous mass. If the process was initiated by acute inflammation, the material is frequently creamy, yellow, and is called pus. They usually occurred in pancreas and brain, which are rich in trypsin and lipid, respectively. This picture displays an infarct in the brain. Here, the tissue is dissolved and turns to a cavity. We also call this kind of infarct as encephalopathy. Under microscope, here and here, we find the pale staining softening foci. The brain cells died and were digested. This picture shows an abscess in liver. The focus is caused by fungal seeding. The focus is here. It is filled with leukocytes and cellular debris. These two pictures are both kidney necrosis. In coagulative necrosis, cells lost nuclei and left clumped cytoplasm. The basic outlines of glomerular and tubular architecture are preserved. In liquefactive necrosis, the dead cells here, they are completely destroyed, replaced by lots of leukocytes and cell debris. The former architecture is totally gone. The third type is gangrene. It is massive necrosis secondary to saprophyte infection. Gangrene includes three forms. Firstly, it is dry gangrene. Limb is the most common site, usually in the lower leg. They are caused by AS or frostbite. This is dry gangrene in toes. The lesion is dry, shriveled, black, and has clear bonder with surrounding viable tissue. Secondly, the wet gangrene. It usually occurs in organs such as lung, intestine, urine, and limb with congestion. When bacterial infection is superimposed, coagulative necrosis is modified by the liquefactive action of the bacteria and attracted leukocytes. The dead tissue undergo leukofaction. This is a specimen of valvulus. The lesion is here, reddish-black colored and swollen, has no clear bonder with surrounding viable tissue. The third, gas gangrene. It happens in opening deep trauma combined with Clostridium perfringens infection. Grossly, the lesion is similar to wet gangrene with the additional presence of gas in the tissues. This is a specimen of muscle with gas gangrene. The tissue is swollen and has honeycomb-like pores in it. That's because of accumulation of gas in dead tissue. Caseous necrosis. It occurs mostly often in tuberculosis infection. Grossly, the lesion here in the kidney is white or light yellow and cheese-like. The texture is soft and is nodular in the cut surface. Under microscope, this area, the normal architecture is completely obliterated. Only amorphous granular debris remained. The fifth form of necrosis, the fat necrosis, it often occurs in the emergency. known as acute pancreatitis. In this disorder, pancreatic enzymes that have leaked out of acinar cells and ducts liquefy the membranes of fat cells in the peritoneum, and lipases split the triglyceride esters contained within fat cells. The released fatty acid combined with calcium to produce grossly visible chalky white area. We call it as fat saponification, which help pathologists and surgeons to identify the lesion. Additionally, in traumatic case, such as memory trauma, can also lead to fat cells destroying and necrosis. This picture shows calcium soap formed in the mesentery. The color of calcium soap is chalky white, like here. These are necrotic fat cells. Some of them still show blurry outlines, like here, and have dotted eosinophilic granules. Here, the dotted granules. The last type is fibrinoid necrosis. It is a special form of necrosis. The common site is fibrous connective tissue and wall of the blood vessel. The lesion is linear or massive red amorphous substance. It's usually caused by malignant hypertension, immunologically mediated diseases such as polyarteritis nodosum, rheumatism. This is an artery in a patient with polyarteritis nodosum. The wall shows a circumferential bright pink area, that is fibrinoid necrosis with protein deposition and inflammation. The deposition includes immune complex and fibrin necrosis. Here. Okay, let's take a summary. We talk about 5 types of necrosis: the coagulative necrosis, the liquefactive necrosis, the gangrene, caseous necrosis, fat necrosis, and fibrinoid necrosis. Here are some questions for you.